everyone, and welcome to today's webinar titled The Evolution from Single Cell to Oncology-Related Pathology. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Sydney McNeil of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by Sartorius. For more information about Sartorius, go to www.sartorius.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speakers, Dr. Andris Abramenkovs, Product Manager, Sartorius, and Mark Lynch, Senior Vice President of Global Sales and Customer Success, BioScribe Genomics. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Andris and Mark, you may now begin your presentation.
Hello everyone, my name is Andris Abramenkovs and I am Product Manager responsible for CellCelector Flex technology. Today, in the first part of my talk, we will discuss the major features of CellCelector Flex platform and then move to discuss single-cell picking process and applications. So what is CellCelector Flex? It is a fully automated system that can isolate cells and colonies from various settings. We can pick adherent and non-adherent single cells, clones, and colonies, as well as larger structures such as spheroids and organoids from a variety of different medias. CellCelector Flex can achieve up to 100% picking and transfer efficiency. The transfer itself is a gentle process that results in high cell integrity and outgrowth. In the video on the right, you can see CellCelector in action, automatically picking cells from source plate and depositing them in a destination plate. The instrument consists of an inverted microscope equipped with a CCD camera, motorized stage holding the cell culture dish, a high-precision robotic arm, multiple racks for consumables, heated holder for plates and reagents, and sterilization vial. The system is coupled with workstation that controls the system itself and stores experiments. CellCelector Flex is an extremely versatile system that can be configured to any picking needs. When configuring the system, it is possible to choose objectives between 2x to 40x magnification to allow identification from a large 3D structure to a single cell. Selection of up to 6 different excitation channels enables support for more complex phenotyping and multiplexing applications. Furthermore, exchangeable robotic arm allows to quickly switch between picking modules to extend the application support. Source vessel is placed in a high-precision motorized stage, which permits to isolate single cells in automated and reliable fashion. Once the cells have been moved to destination plate, this plate can be heated or cooled based on the needs. For example, for omics experiments, sample cooling during extraction is crucial, as keeping DNA, RNA, and protein cooled ensures better stability of these structures. When looking closer at the picking modules, the system can be equipped with semi-solid picking module that takes advantage of disposable tips, adherent colony picking module that is using reusable stainless steel tips, and a single cell picking module that is using glass capillaries and is the most versatile picking module. Once the picking module is installed in the system, it is automatically recognized and all corresponding picking protocols are loaded within the software. When it comes to picking and depositing of cells, CellCelector Flex allows to pick almost from anything to anything. As a source vessel, we can use nanowell arrays, cell culture microplates with clear flat bottoms, microscope slides, petri dishes, and even custom plates. We can deposit the material in cell culture microplates, multi-well slides, petri dishes, and even PCR tubes, strips, or plates. With extended support of different vessels, we can extract cells from cell culture and tissue samples, like tissue sections and everything in between. Using our cooling deck tray, we can make sure the destination vessels are always cooled, making sure that the extracted material is protected from degradation. The CellCelector is all-in-one solution that guides users through steps of scanning, object detection, review, and transfer of cells. After scanning of a plate, the object can be automatically detected according to user-defined parameters. Then, selected objects can be further filtered, sorted, and transferred to a destination plate. During the experiment, overview images, numerical data of all cells, and QC images are automatically saved in the database and can be exported for reporting. All of the objects identified in analysis step has a unique ID assigned throughout the experiment and can be easily traced. Before and after picking images, ensure that you have picked the cells of interest and nothing more, nothing less. Optionally, automatic assessment of picking success is also available. In the second part of the talk today, I will focus on single-cell applications and isolation. So why single-cell analysis is important? It allows us to identify cell heterogeneity within tissues by allowing to characterize each single cell based on their epigenetic modifications, genetic properties, gene expression status, and metabolic state. With traditional bulk analyses, this heterogeneity might go unnoticed, especially if some subpopulations are low in numbers. In disease research, single-cell analyses can help to separate diseased cells and help to identify key genes and pathways driving disease progression. Further, these results can be used to discover new drugs and biomarkers. Alternatively, information from single-cell analyses can help to identify molecular signatures that are unique to patient's disease. This can then be taken further to develop tailored treatments. In developmental biology, single-cell analyses can help to understand how different cells are arising during development and how they are differentiating into specialized cells. Now we're going to focus more on the single-cell picking on CellCelector. CellCelector's single-cell picking module is a versatile tool that can support many applications, starting from purification of single cells for omics experiments to isolation of small tumoroids and organoids. Please note that there are more applications supported than listed here. So how do we achieve the large application support with CellCelector? The answer lies within hardware capabilities of our single-cell picking module, which can hold wide variety of glass capillary diameters that allow to pick single cells, clusters, and even small organoids or tumoroids. Also, high-precision aspiration ensures gentle cell retrieval and preserves cell integrity. The tight control of aspiration volume allows us to pick up a single cell without disturbing neighboring cells. Moreover, pickup of cells from different plate formats can be calibrated in software by using extremely precise touch sensor inbuilt in robotic arm. When using a single-cell picking module, it is possible to take advantage of nanowell arrays that support adherent and suspension cell isolation and culturing. With this module, single-cell retrieval takes about 25 seconds, while pooling of single cells takes about 10 seconds per cell. CellCelector supports many different single-cell capture formats. Cells can be extracted from standard plastic plates or dishes. For suspension cells here, the cells need to be immobilized on the surface of a plate to restrict the cell movement during scanning and picking. As an alternative, cells can be also picked from magnet pick slides that are using magnetic field to align the beads and keep the cells in their position for screening, detection, and picking. In this approach, cells have to be pre-processed using positive immunomagnetic enrichment methods. Lastly, single cells can be isolated using our nanowell plates that can capture one cell per well, and once all of the cells of interest are detected, they can be easily retrieved. Here we will be looking at some of the examples of single-cell isolation from standard plastic surfaces. Even highly adherent endothelial cells are possible to be isolated. Please note that in this example we used adherent colony picking tool with stainless steel tips. Adherent cells can also be extracted using single-cell picking tool as shown here with example of HeLa cells. You will quickly notice that these two cells are very closely located, even though we were able to retrieve one of these cells without disturbing the other, just illustrating the extreme precision what is possible with CellCelector Flex system. When it comes to non-adherent cells, the isolation process is more challenging because without any coatings like poly-L-ornithine or poly-L-lysine, the cells can shift focal planes and move around during scanning and retrieval process. When we are using coatings to immobilize the cells on the surface of the plastic, we need to perform additional tests to make sure that the cell biology is not affected by the coating itself. In the example here of non-adherent cell picking, you will see a mouse CD4 positive T cells and HT29 cell isolation. The positive immunomagnetic enrichment starts with a heterogeneous sample in solution. The specific population of cells are labeled with antibodies and magnetic particles. Using magnet, the successfully labeled cells are retained in the solution while negative cell population is washed away. Then cells are stained for further characterization using antibody cocktail, and afterwards the cells are captured on our magnet pick slide and transferred to CellCelector for scanning, analysis, and isolation. Single cells then can be analyzed based on their DNA, RNA, and protein contents. Usually this kind of workflow is used for extracting rare cell populations, like for example, circulating tumor cells. In the example here, we have stained circulating tumor cells with DAPI and two different cell surface markers from which one has to be positive and the other one has to be negative for cells to be classified as a circulating tumor cell. Once all circulating tumor cells have been identified, the cells can be isolated and sequenced. Overall, isolation of cells using positive immunomagnetic enrichment is a reliable method for isolation of single cells when paired with our magnet pick slides. However, it is the staining procedure with beads and antibodies that is time-consuming and costly. As we discussed before, single cell isolation can be a tricky process, especially for cells in suspension, as we need to ensure that cells are not moving around during imaging and picking. This is where our nanowell plates come into play, as they allow to restrict single cells to each nanowell. These nanowell arrays are available in plate and slide format, and the size for each individual nanowell ranges between 25 microns to 200 microns. Nanowells with smaller wells can be used to isolate single cells, where larger nanowells can be used to expand and establish new cell lines. Nanowell plates come also in different shapes and are available in square, hexagonal, and elongated shape. Based on the format, it is also possible to select up to 200,000 nanowells per well. Plating cells in a nanowell plate is easy and takes only 5 minutes, as we add cells in the media to well and spin them down using centrifuge or let them sediment. These plates support high throughput experiments and save time and money in the process. And mainly that's achieved by replacing stacks of 96 and 384 well plates. In this example, we will be looking at a single well from U25 slide that contains 200,000 nanowells per each individual well. The slide here is scanned with 10x objective and it is acquired in bright field channel. All of the individually acquired images are stitched to create an overview image of whole well. Using high quality imaging platform, we can easily identify cells when zooming in on each individual nanowell. Even with a 10x objective, we can also see individual beads. After completion of scan, we can move forward to analysis. User-friendly analysis tab allows us to quickly generate analyses for each individual channel. We can select multitude of different analysis parameters for characterization of your cells. For example, size, shape, sphericity, fluorescence intensity, and many more. Each measurement comes with a description so that we can easily understand what each number really means. It also helps when additional filtering and processing is needed. Once the analysis definition is created, we can save it and use it for our next experiment. When analysis is completed, each identified cell or object is assigned a unique ID for traceability throughout the experiment. This makes sure that no cell gets lost during the process. Then, in the next step, we move to the filtering, and it's possible to filter all cells based on the parameters selected in analysis. For example, like morphological properties and fluorescence marker positivity or negativity, which allows us to narrow down to identify the objects of interest. All filtered objects then are listed in a gallery view where we can inspect them visually. From the gallery view, we can also manually deselect objects to remove them from the pick list. Now it's time to move to picking procedure, so let's talk about it in more detail. Before picking cells, we can pre-fill capillaries with buffer solution from buffer station or destination plate. Then we can pick single cells and deposit them in destination plate. After finishing the picking procedure, the capillary can be sterilized as needed. This workflow is reiterated until all of your cells of interest have been transferred. The workflow here is just an example, as various routines can be programmed to ensure the capillary tip is free of carryover material from sample to sample or single cell isolation to single cell isolation. In the video here, you will see a single cell picking process in real time, and you can monitor this process at any given time during cell picking. You will also notice in this video that some of the frames will be flickering. This is the time when we are actually acquiring images. So you will notice that we acquire images at the beginning of the picking procedure and then at the very end. So once the picking has been successfully completed, we move to the next cell, acquire image, take out the cell, and then acquire image again. This ensures that we have a proper documentation throughout the whole experiment. In summary, CellCelector is a truly versatile system that supports many different cell isolation setups. It also makes cell isolation easy, preserves cell integrity during the cell picking process, and assures proper documentation. CellSelector is a custom configured to your picking needs, and please do not hesitate to reach out to us if you have any further questions. With that, I would like to thank you for tuning in and looking forward to your questions in Q&A session. Now that we have talked about how to quickly and easily identify, select, and retrieve a single cell with the CellSelector platform, I'll hand over to Mark, who can take us through how these single cells can be used for low-input single-cell genomics.
Thank you. Today, I will introduce to you BioScribe Genomics' unbiased whole genome and whole transcriptome chemistries for deep characterization of single cells important in cancer heterogeneity studies. There are two chemistries to introduce to you in this webinar. The first, Resolve DNA. Resolve DNA provides unprecedented genomic coverage and uniformity for calling of single nucleotide variation, copy number variation, insertions and deletions, and somatic mutations. The second, and perhaps most important in the era of multi-omic analysis, is Resolve-Ohm. Resolve-Ohm takes the same Resolve DNA chemistry and adds in full-length cDNA analysis. Today, we will therefore inform you on technology for whole genome and whole transcriptome analysis from the same single cell. Let's start with Resolve DNA. Resolve DNA is the core of our technology at BioScribe Genomics. Resolve DNA is powered by a method known as PTA, Primary Template Directed Amplification. Resolve DNA with PTA methodology increases the fraction of the genome covered, provides much higher uniformity of the genome, and a much better allelic balance than any other method of whole genome amplification on the market. The first way in which Resolve DNA does this is by using multiple random primers. These are shown on the schematic on the right-hand side of the slide. These multiple random primers also have terminator molecules added to them. You can see these in purple, also in the schematic on the right-hand side of the slide. These terminator molecules terminate the random primers after a short period of extension, normally between 250 and 1500 bases. This termination then enables the extension of the next random primer and termination of that next random primer. And it continues doing that until the reaction is complete. What this creates are short fragments, and these short fragments do not re-amplify the previous fragment. Instead, that primary template is reprimed for what we call additional favored amplification. In other words, it's directed back to the original DNA template, creating this favored amplification. The random primers, the terminators, and the favored amplification ends up creating a short fragment of DNA that is around 500,000-fold amplified. And because we use a quasi-linear based amplification protocol, we generate excellent uniformity, coverage, and reproducibility. And so these 500,000-fold amplified DNA products are covering more than 97% of the genome. This is very important in characterizing tumor heterogeneity. The standard workflow for Resolve DNA looks a little bit like this. Cells are sorted into a microwell plate, such as a 96 or a 384 well plate. This can be achieved using flow cytometry, or it can be achieved using the CellCelector system from Sartorius. Once those cells are in those 96 or 384 well plates, there are two simple steps that the Resolve DNA chemistry takes care of. This is shown in dark blue on the right-hand side of the slide. The first is the cell lysis step, and the second is the whole genome amplification step. These two critical steps take just 30 minutes hands-on time. And once it's complete, the BioScribe Genomics library preparation workflow generates libraries for processing on next-generation sequencing platforms. And once the data is complete, the data can be analyzed using the BioScribe Genomics Base Jumper Analysis Suite. In summary, the workflow for Resolve DNA is end-to-end, with the exception of the sequencing piece in the middle. An example of how our Resolve DNA chemistry is used is in the field of cancer heterogeneity. And an example of this of particular importance is in something called ductal carcinoma in situ, or DCIS. DCIS is a neoplastic proliferation of ductal epithelial cells that can be a precursor to invasive breast cancer. On the schematic in the slide, you can see how this develops from normal into hyperplasia, into atypical hyperplasia, and then eventually into ductal carcinoma in situ, or DCIS. A fundamental research mission is to understand the influence of the stromal microenvironment and cell and genomic events that drive this transition to invasion. Using the Resolve DNA chemistry, the comparison between the cell lines, in this case GM12878 cells, and ductal carcinoma in situ cells is very, very close. We are recovering with Resolve DNA more than 95% of the genome in most single cells. And importantly, this coverage of uniformity is happening in primary sample as well as cell line. Looking at the copy number variation plot and the CNV plots on the right-hand side of the slide, you can see that with normal, we have a kind of normal CNV plot with no aneuploidy. The chromosome structure was found to be intact. When we look at cell 1, cell 2, cell 3, and cell 4, shown as B, C, D, and E, you can see the differences in the copy number variation plots. The tumor 1 sample, labeled B, looks pretty normal. But when we get to tumor 2 and tumor 3, we start to see some of the deletion events in chromosome 13, 16, and 17 that are known to be associated with breast cancer. And when we get to tumor 4, labeled as E, we actually identify other deletion events with the Resolve DNA chemistry in chromosome 11 that were not expected to be seen. And as part of our collaboration in this, we were identifying new and unexpected actionable genomic information, meaning that in this particular individual, this cell and these cells enabled a change in the treatment regime of that particular subject. Resolve DNA is therefore an isothermal quasi-linear method that delivers unbiased, uniform allelic balance, coverage, and reproducibility to deliver a complete and comprehensive view of the single-cell genome. The chemistry is compatible with upfront cell selection and cell sorting methodologies, offering flexibility. And it's a simple and refined workflow with no expensive single-cell instrumentation needed. And it's scalable. Our chemistry can be run in 96 or 384-well formats. The second chemistry to introduce to you today is Resolve-Ohm. Resolve-Ohm enables comprehensive single-cell multi-omic analysis. Many of you will ask the question, why study the genome and the transcriptome from the same single cell? The reason is that there is an interplay between the RNA and the DNA, and that information helps the diagnosis and understanding of pathogenesis and cancer susceptibility. So if one is studying tumor heterogeneity and trying to correlate heterogeneity to pathogenesis, single-cell multi-omic analysis is the only way to do it. A publication in 2022 from Horton et al. Actually stated that DNA sequencing alone is limited in its detection and classification of mRNA splicing variants, particularly those located far from the coding sequences. As a result, Resolve-Ohm is the only method in the single-cell field that provides heterogeneity, a correlation to pathogenesis, and a full characterization of single cells. The schematic in this slide actually shows that even droplet-based methodologies cannot deliver the multi-omic analysis features in the same way that Resolve-Ohm can. And in fact, Resolve-Ohm from BioScribe Genomics provides more than threefold data per single cell than droplet-based methods. The workflow is very similar to a Resolve DNA workflow that we covered earlier in the presentation. The key differences really is the lysis. The cells are collected in the same way, such as using flow cytometry or by using the CellCelector system in 96 or 384 well plates. And once those cells are collected, we perform a cytosolic lysis to permit the first generation and first strand cDNA synthesis to take place. Once that's complete, in the same well, the Resolve-Ohm chemistry then performs a nuclear lysis to disrupt the nuclear membrane to enable the amplification of the genome. And so all of this is taking place in the one well. The cell is not split at all. Once we've amplified our whole genome, it is at that point during the library preparation that the cDNA arm of Resolve-Ohm and the genomic DNA arm of Resolve-Ohm is split. And each of these, shown on the right and shown on the left, undergo parallel library preparation using the Resolve-Ohm library preparation kit. Sequencing is then performed on sequencing platforms, and data analysis is performed using the base jumper solution from BioScribe Genomics. The transcript data that's generated from Resolve-Ohm is shown on the left-hand side. The first thing to note in this column is that the full-length detection of transcriptome features is only possible because Resolve-Ohm is using a full-length cDNA analysis, including the 5' and the 3' UTR. So we overcome the bias observed by droplet and end counting methods. The second thing to note is that we are detecting around 7,000 genes per single cell. 7,000 genes per single cell. It means that we're detecting a lot more information than is possible using droplet-based methods. And we're able to analyze splice variants, fusion genes, and transductions. It's also important to note that we do not get any genomic contamination in this workflow. The library preparation and the sequencing of the genomic arm, as well as the mRNA arm, are absolutely separate, and there's no crossover between the two. Resolve-Ohm is the multi-omic solution for detecting DNA and RNA from the same single cell. And because this era of multi-omics is so important, our plan is to start to include targeted protein analysis within the Resolve-Ohm chemistry. More information can be found on our bioRxiv paper. To summarize the Resolve-Ohm, it's a broad single-cell multi-omic analysis solution that combines the Resolve DNA primary template-directed amplification with full transcript reverse transcription. It's comprehensive in its depth of information, with more than 90% of the genome and full-length mRNA coverage revealing the consequences of genomic variation on gene expression and transcript structure. And it's an accurate and unified workflow that is simple and easy to use, interrogating DNA and RNA without splitting source material or interpreting across multiple data sets. It's scalable and readily available with 96 well plate formats without the need for expensive droplet instrumentation.
Thank you for your time and attention today. We are happy to have been able to share with you this integrated workflow enabling spatial single-cell genomics. You can find the full application note on our website, but we also will make sure to share this on this platform for all attendees. And now, we're happy to take your questions.
All right. Thank you, Andris Abramenkovs and Mark Lynch, for your informative presentation. We will now start the Q&A portion of the panel. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. Okay, our first few questions are for Andris. Number one, what are the advantages of CellCelector and why is it so unique?
So CellCelector is a unique technology that combines a powerful high-content imaging system and fully automated self-picking robot. Generally, the system has very quick scanning and picking speeds that are essentially gentle to the cells as well. So the combination of unique features also gives the highest cell outgrowth rates almost in the whole industry. And also it allows you to detect, select, and isolate single cell clusters, spheroids, organoids, tissue fragments, adherent colonies from liquid media and semi-solid media. And this flexibility is really what sets CellCelector apart.
Okay, wonderful. Thank you. Our second question is, can you talk about the CellCelector nanowell arrays and how they are used?
So CellCelector nanowells come in a size from 25 micrometers to 200 micrometers in diameter. So essentially the number of nanowells can be anything between 3,000 to 200,000 per well. So usually the smaller sized nanowells are used for separating single cells, while the largest size nanowells are also used for not only separating the single cells, but also allowing the cell expansion for example, cell line development experiments.
Perfect. Thank you. Our next question. Why were the chips designed in the shape they are?
So the spindle-shaped nanowells has a round middle area that allows more easily separate suspension cells, meaning one single cell per chip. By matching the size of the object, it is also much easier to ensure that really we are fitting one cell per well. While if you're having for example, a larger area, like for example, square shape, the diameter is actually measured in diagonally, which means that we can essentially fit more cells per well and also expand cells if desired in these kind of shaped chips.
All right, perfect. Next question. What is the final volume of CellCelector and can it be controlled at low volume?
So in general, the maximum volume that we can transfer using a single cell tool is about 12 microliters and minimum is 1 nanoliter. So essentially anything in between the range you can select. With adherent picking tool, we can extend the maximum range up to 32 microliters and semi-solid media picking tool up to 20 microliters.
Okay, wonderful. It looks like we have one more question for you, Andris. What are some of the other applications for CellCelector?
Oh, yeah, absolutely. Besides isolating single cells, CellCelector can be used for cell line development to transfer productive cells and well-growing clones. We can also use adherent stem cell colony for subpassaging and analyses. We can use CellCelector for rare cell isolation and circulating tumor cell extraction. And also we can extract larger objects such as spheroids and organoids from 3D matrices and deposit them also in the 3D matrices.
Perfect. Thank you so much, Andris. Our next set of questions here is for Mark. First question, how does the CellCelector help the BioScribe workflows?
Yeah, so the CellCelector system enables the users to select cells from within tissues. And in the single cell multi-omic field right now, this is a really important area in terms of spatial analysis of how the cells interact within a tissue. And so the reason for partnering with Sartorius is actually to be able to use the cell selector to specifically target cells of interest, remove those cells of interest from the tissue, and put them directly into the BioScribe genomics chemistries and workflows. So really it helps by going straight from the tissue right into the molecular workflows of BioScribe.
Great, thank you. Our next question is, in the Resolve-Ohm technology, is the cDNA amplified? If not, why not?
That's a great question. So the Resolve-Ohm technology, as mentioned, is BioScribe Genomics multi-omic single-cell technology. The idea behind it is that we take RNA and DNA from the same single cell. Now, because you're starting from a single cell, it is important to amplify the cDNA, which is the transcriptomic part, and the genomic DNA, which is the genomic part. And so to answer the question directly, the cDNA is indeed amplified in Resolve-Ohm. The trick, however, in the workflow is being able to produce a chemistry like Resolve-Ohm that amplifies the cDNA without it impacting the genomic DNA. And so as part of the workflow, there's a step by which you take that amplified cDNA and pull it away from the genomic DNA. And so you get a very clean pool of cDNA amplified and a clean pool of genomic DNA amplified. So to answer the question, it is amplified in that workflow, but it's done so in a very clever way.
Wonderful, thank you. It looks like we have one final question for you here, Mark. When do you split the cells during the Resolve-Ohm multi-omic workflow?
Yeah, it's another good question. So actually, the neat part about it is that we do not split the cells in Resolve-Ohm. We split the nucleic material that's amplified later in the workflow. The reason that's important to note is because you start with a single cell in a well, for example, using the cell selector technology to pick those cells and put them into a microwell. And that same cell undergoes the reverse transcription and it undergoes the genome DNA extraction step at the same time. It's done in the one well. So we actually don't split the cells. That's really important to make sure you're getting the transcriptomic and the genomic signature from the same single cell. But later on in the workflow, we do split it. We split the cDNA amplified material and the genomic DNA amplified material so that it's then ready to go on a next generation sequencer. And it's at that point we split. But it's very important to note we do not split the cells.
Great, thank you so much. Thank you again, Dr. Andris Abramenkovs and Mark Lynch. Do you have any final comments for our audience?
Thank you for joining in. And of course, if you have further questions, please do not hesitate to reach out to us and we will be more than happy to get back to you. All right, thank you.
All right, thank you both again for your time today and for your important research. Before we go, I would like to thank the audience for joining us today and for their interesting questions. I would also like to thank our sponsor, Sartorius, for sponsoring today's webinar. Questions we did not have time for and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. If you would like to find out more about CellCelector Flex, visit the Sartorius booth in the virtual exhibit hall to access further information and resources to learn how this fully automated and uniquely versatile system can meet cell imaging and retrieval needs. This webcast can be viewed on demand for 2 years until May 18th, 2025. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.